Hello, everybody. My name is Boro Dropulić. I'm the Executive Director of Caring Cross. Welcome to our virtual seminar, where today we are joined by Dr. David Williams, a pioneer in the gene therapy field, and he was also Chief of the Hematology and Oncology at Boston Children's Hospital. He is also the President of the Dana-Farber Cancer and Blood Disorder Center and the Leland Fikes Professor at Harvard Medical School in Boston. Welcome, David. Thanks for joining with us.
Thank you, Boro. Happy to be here.
Wonderful. And we are certainly grateful for your time. So everyone, Dr. Williams will provide a clinical update on exciting data using his lentiviral vector-based shmiR stem cell-based therapy for sickle cell disease that he has been developing with his team members over the number of years. After the talk, right after the talk, I'll give a brief overview of the partnership between Caring Cross and Boston Children's to deliver these therapies for underserved populations, especially those in low middle income countries. And before we get started, just one administrative note. If you would like to ask and pose questions, there's a box there, a Q&A box. You can submit your questions and we will be able to answer them then right after the talk. So without further ado, David, over to you.
Thank you, Boro. I'm grateful for the opportunity to talk about our technology. Let me share slides and put this on slideshow. And so what I'd like to do is give you all a background on sort of the biology behind which we sought to develop the technology that we did, and then talk a little bit about that technology itself, cover our first in human trial, which is concluded, and I'll touch base on the pivotal phase 2 trial, which is now closed to enrollment and has all patients infused at this point. And this all has to do with this figure here, in which we're pointing out that our technology has as its basis elevating fetal hemoglobin in red cells and concomitantly decreasing hemoglobin S, or sickle hemoglobin. So here are my conflicts or disclosures. Sorry, go back. And I'll just point out two. One is that the old Bluebird, it's not called Bluebird anymore, provided the GMP vector from our plasmid, basically, for the clinical trial from a previous agreement. And then, as mentioned already, we've just licensed the vector and the trial data to Caring Cross. So a little bit of a background here. Let me get rid of, if I can do it, I guess I can't. A little bit of background on sickle cell disease. You probably are all aware that it's a molecular disease caused by a mutation that leads to an amino acid change at position 6 of the beta globin subunit of hemoglobin. And this leads to an abnormal molecular interaction that has fibrils formed in low oxygen concentration. And those fibrils have many different pathophysiologic results, one of which is the sickling of the cell, as you can see here, that the disease gets its name from. But the clinical manifestations are really manifest in the whole body. There's chronic hemolytic anemia. Probably the most problematic thing for most patients is pain, both acute and chronic, and end organ damage, including stroke, hypertension, renal disease, avascular necrosis of the femoral heads in particular, and stasis ulcers, particularly in adults. Now, this is one of the most common genetic disorders worldwide, which is why we're quite excited about our collaboration with Caring Cross. There's about 1,000 births in the US each year, but worldwide there's probably more close to 300,000 births a year. And in the low-income nations, many patients with this disease die early in their lifetimes. And the estimate of what's going to happen in the future, as you can see here, is a large increase in the number of patients that have the disease. And because in developing countries, there's now increasingly use of penicillin prophylaxis, many more infants are going to survive into adulthood. This is also a tremendously burdensome disease with respect to the economies of countries. For instance, in the United States, where there's thought to be about 100,000 patients with the disease, it's estimated that there's about $1 billion a year in healthcare charges. Now, there has been progress in this disease. So here is percentages of death with time in different age groups. And you can see that there's a dramatic drop in deaths under the age of 5. This is completely due to newborn screening and the institution of penicillin prophylaxis in patients from early age. But you can also see, while there's been some shift in the percentage of death to a higher age in adulthood, but this you can see that the life expectancy for patients with sickle cell disease in the United States is still much shorter than individuals without sickle cell. So there's been progress, but certainly there needs to be more progress, of course. Now, there has been, as you know, different approaches now to genetic therapies for sickle cell disease, 2 of which have been approved by the FDA in the United States. One is transgenic expression of either a gamma globin gene or a mutated beta globin gene that acts like gamma globin to be resistant to sickling. This is the Bluebird approach, now genetics biotherapeutics. There's transgenic knockdown of BCL11A, a repressor of gamma globin that I'm going to go into much more detail on to induce fetal hemoglobin. And that can be done with gene editing to remove an enhancer sequence that regulates the expression of this transcription factor in red cells, work done by Dan Bauer and Stu Orkin's lab. There's also work to generate what's called an HPFH mutation of the gamma locus. This is hereditary persistence of fetal hemoglobin. So these are individuals that have mutations in the gamma locus that resist being turned off, and therefore they have very high expressions of fetal hemoglobin in adult life. And there's even programs in biotechnology to actually repair the sickle cell mutation itself. Now, why is there a focus on hemoglobin F in sickle cell disease? So there's 3 studies often cited, 2 from Ora Platt, who's here retired with Boston Children's, where either measuring pain or acute chest syndrome, which is a complication of sickle cell disease, or death, you can see in these indications or consequences of sickle cell, the higher the hemoglobin F you have, the less frequent the pain episodes, the less frequent acute chest, and the longer your lifespan is. So even this change of hemoglobin F, I just want you to remember the numbers of 9% below or above for fetal hemoglobin has a fairly dramatic effect on adding another decade to life in patients with sickle cell disease. So it's been known for a long time that fetal hemoglobin mitigates sickle cell disease phenotypes. But what is fetal hemoglobin? This is a diagram that shows the hemoglobins that your red cells express throughout life. And you can see here that in utero, on the left of this birth mark here, you express alpha globin genes, but you express, instead of beta globin genes, you express gamma globin genes. So that makes up fetal hemoglobin, so alpha and gamma. When you're born, the bone marrow red cells shut off expression of gamma globin and turn on expression of beta globin. And of course, that means early in life, if you have sickle cell disease, you have no symptoms. But later in life, when this switch occurs and you start expressing sickle hemoglobin as your beta locus turns on, then you develop symptoms. Now, the other thing about fetal hemoglobin is a very effective hemoglobin to resist sickle hemoglobin polymer formation. So this is a very old study from the NIH, but quite informative. So along the x-axis is oxygen concentration. On the y-axis is polymer formation. So you see here a patient who has sickle trait, and you know, in general, sickle trait has no symptoms. And here's a patient who has hereditary persistence of fetal hemoglobin co-inherited with sickle cell disease. And the interesting thing about these is these patients are asymptomatic, but this patient's red cells who has trait has 41% sickle in their red cells, and this patient has 63% sickle. So this difference shows you how effective fetal hemoglobin is versus normal hemoglobin A in resisting sickling in the red cell. So we know that the more fetal hemoglobin you have, the more attenuation of sickle cell you can see. So for instance, there are mentioned patients that co-inherit HPFH. There are high F populations in Saudi Arabia and in South Asia who have a milder phenotype, and good responders to the drug hydroxyurea have less clinical symptomology. And you can see in this review by George Buchanan, which is now quite old, that he hypothesizes that your fetal cells, your F cells, the cells containing fetal hemoglobin, need to contain about a third of the hemoglobin as hemoglobin F to try to retard the sickling phenotype. I just want you to remember that number too as we go forward. So this led for many, many years, for decades, to NIH funding and research to try to understand what's called the fetal to adult switch. So why does the bone marrow choose to shut off expression of fetal globin after you're born and turn on the adult beta globin? And the breakthrough in this area came from GWAS studies shown here. Those of you that know these studies, these are called Manhattan plots. And you ask the question, what are the gene sequences that are associated around the world with thousands of genomes with a particular phenotype? And in this case, the question was with high fetal hemoglobin. And you can see, surprisingly, on the left-hand side here, a gene that popped out that no one would have predicted at the time, called BCL11A. This stands for B-cell leukemia 11A. So this was a transcription factor known by immunologists to be required for normal B-cell production and known in oncology because it's mutated in some patients with leukemia. And a seminal work in Stu Orkin's lab, shown here and published, I will go very quickly through this, shows that in the fetal liver, there's very little of this expression of this gene. But in adult bone marrow, there's a much higher level of BCL11A. And what's more, if you use an shRNA to knock down BCL11A, you can see in this middle bar here on the right that the knockdown with an shRNA leads to very high level now of reinitiation and re-expression of gamma globin. So in essence, the diagram shows when you have BCL11A around, you turn off gamma globin and you turn on adult beta globin. So basically, we understood this basic science data and hypothesized that this would be a very good molecular target for sickle cell disease. And we call this flipping the switch on sickle cell disease. So the idea is to turn back the clock in individual patients' red cells so that instead of having beta S expression, we manipulate the BCL11A complex and then reinitiate expression of fetal hemoglobin in the cells as a therapeutic target, so that we simultaneously increase fetal hemoglobin. And because we're using a physiologic binary switch, we concurrently decrease the expression of hemoglobin S. And since the sickling in a red cell is exquisitely dependent on the concentration of hemoglobin S, you basically get a twofer for this one switch. You get more resistant expression of fetal hemoglobin, and you get decrease in hemoglobin S in the cell. So our goal was to rapidly translate these findings into sickle cell disease using currently available viral vector platforms. So approach was to knock down BCL11A via RNAi using lentiviruses to induce gamma globin expression and silence mutated beta S expression. And we call this post-transcriptional gene editing. And the advantage, as I mentioned already, is that the sickling beta globin gene is downregulated in proportion to and simultaneously to increased expression of fetal hemoglobin gene. Now, it turns out that wasn't so easy to do as it is in the laboratory. We had to solve two biological problems. One is, you may know or you may not know, but I'll say that high levels of expression of antisense RNA is toxic to cells. This is a non-specific toxicity, but related to the expression of antisense RNA. And because BCL11A is required for B cells development, and we discovered in our translational work in this project that it's also required for hematopoietic stem cells to function. This wasn't known previously. You need to knock down BCL11A only in red cells, not in B cells and not in hematopoietic cells. So to do that, we had to develop a new type of vector we called a shmiR, where we basically embedded the shRNA in red here to BCL11A in a microRNA scaffold, so that the cell actually processes this as an endogenous microRNA. This is sort of shown schematically here, where you can see the shRNA sequence in blue embedded in a microRNA processing. And this mimics microRNAs. It's more physiologic, and therefore there's less non-specific toxicities. And it allows us to regulate the expression only in erythroid cells, avoiding the toxicities of stem cells and B cell deficiency. So this is just one slide from our IND application, where we use the sickle cell mouse model called the Berkeley mouse. We use the vector to put the shmiR into the red cells, into the stem cells, and then we transplanted these sickle stem cells back into normal boy J mice that have been irradiated. And so the salient data is shown below. And by flow, you can see in a control mouse that there's a very high level of circulating red cell precursors indicative of the sickle cell phenotype. And as opposed to that, when we use our shmiR shRNA, that level of primitive red cells goes down almost to the level of wild type. So this was quite dramatic phenotype correction. There was a lot of other data that we did for the IND, but that's an important one. And then this is the second, which is 3 different patients with sickle cell disease, where we took their bone marrow stem cells, transduced them with the human clinical version of the vector. This is, you can see referenced down below. And if you look on the left, this is induction of fetal hemoglobin by either protein or RNA. And you can see in triplicates in all 3 patients, there's very high level induction of fetal hemoglobin. This is a mean of almost 80% fetal hemoglobin. We'll come back to that number in a little bit. And then on the right, you can see simultaneous reduction in hemoglobin S from the usual 80% in these patients down to less than 20%. So this was a key experiment in the IND to allow us to move forward for human studies. So this is the basic study designed for lentivirus ex vivo gene therapy. The stem cells were mobilized in patients with sickle cell disease into the peripheral blood and collected by apheresis. Their CD34s are selected in a GMP lab, transduced and cryopreserved, and then a number of safety tests are done for the product before it's released. Patients admitted to the hospital given conditioning, myeloablative conditioning, busulfan, and then the cells are infused and then patients are followed. So this was a first in human ever to receive a shmiR vector, and this has been reported in the early phase, as you can see in the New England Journal of Medicine, which really was the validation of BCL11A as a therapeutic target in sickle cell disease in humans. Now, just to show you some of the data emanating from that initial study, we actually proved that the vector worked by plating the product, the gene therapy product before infusion into colony assays where we looked at red cell colonies called BFUE and did HPLC on individual colonies and asked the question, do these individual colonies show enhancement of hemoglobin F in red cells? And you can see at the top, the percentage of BFUE having greater than 30%. Remember, I mentioned this percent level in my preliminary discussion of the disease. So our release criteria for the product was that we had to have at least 50% of these red colonies with greater than 30% hemoglobin F. And you can see in all these patients, not only did we surpass that 50%, but we really surpassed it in great amounts. And you can see the blue dotted line where the 30% is, and you can see all the colonies that are above that 50% or 30% mark. So that initial pilot study enrolled or infused 10 patients. We now have follow-up almost 6 years. The vector is particularly safe. There's been absolutely no adverse events related to the vector itself. And it works extremely efficiently to transduce hematopoietic stem cells. I'll come back to that. The expression is lineage specific, so it did what we wanted, only in red cells, no expression in B cells, no expression in stem cells. And the patients have very stable gene marking over now years. The F is induced broadly. I'll show you a little bit of this data in a minute. And importantly, to get back to the review by George Buchanan, there's a high hemoglobin F for F cells, so over 10 picograms of F, or a third of the hemoglobin in these cells are hemoglobin F. And there's been significant attenuation of the Sickle phenotype. Now, this was a phase 1 study. It wasn't designed for efficacy, but we did collect some efficacy data that let us move to the phase 2 study. And importantly, we used this study to do dose finding. So we manipulated the manufacturing parameters with subsequent patients so that we could optimize the transduction to the highest level. So this led then to the pivotal phase 2 trial. This is an NIH-supported and CIRM-supported trial, California Institute of Regenerative Medicine. And this was a trial in which we negotiated the endpoints with the FDA so that if we hit our endpoints, this data could be used for a BLA submission and drug approval. The name of the study is GRASS. It stands for Gene Therapy to Reduce All Sickle Pain. And the trial opened in 2023. Unfortunately, it was put on hold for almost a year by the NIH, the funder, not the FDA. We had 9 sites. 32 patients were enrolled, 28 passed screening. We targeted 25. There was 1 mobilization failure and 1 patient withdrew. We validated a second lot of GMP vector and introduced clinical transduction that was optimized from the phase 1 study. And 26 patients have been infused and 25 are evaluable. There's been no safety signals to date at all. And we had additional genomics safety surveillance as dictated by the NIH, where both pre and post infusion, we monitored genes that are associated with myeloid leukemia or MDS. This was due to the leukemias that occurred in the Bluebird trial. And this included an expert independent genomics panel adjudication panel that looked at the large amount of data that generated from screening all these genes. No pathogenic mutations been identified in any patient. So I'll just quickly end with some of the data from the phase 2 trial to give you a sense for how good the vector works. So here's patients listed down the left-hand column, and I would just draw your attention to the percent BFUE with greater than 30% hemoglobin F, and you can see how effective this vector is. So the range of that number is from 54 to 99% effective, which is well above our release criteria. And probably more importantly, here are the data, 6-month data on the first 19 or so patients. I think it's actually 15 patients that were infused. And the 2 columns to pay attention to are the percent F cells. So that's the number of circulating red cells in the blood at 6 months that have fetal hemoglobin. And you can see that is 86% or 87%. So the vast majority of red cells have detectable levels of hemoglobin F. And also, if we just do electrophoresis on the peripheral blood, you can see quite a dramatic induction of fetal hemoglobin in the peripheral blood. So the average of these patients is 44% F. So just remember my introduction where Oraplat showed that above 9% made a huge difference in the lifespan of individuals. So you can see that this is quite a significant induction. And we're quite optimistic. We don't know we're blinded from the efficacy data at this point, but we're hopeful that this is a transformative therapy as we think it will be based on the phase 1 study. So just another point, this is a pivotal trial. So I mentioned we have an independent appointed genomics committee. We have an independent appointed VOE adjudication committee. We have an independently appointed DSMB. There's independent clinical monitoring and auditing by EMIS, and we have independent auditing of the manufacturing process. This is all just to point to the fact that this is a very rigorously run and robustly monitored trial. And so, as I said before, we don't know what the clinical outcomes are at this point, but we're very optimistic that they're going to be good. So moving forward, in the United States, we are developing an amendment to the IND called an expanded access amendment, which, if you look down below, will hopefully open gene therapy to patients with hemoglobin SC disease. These are patients that haven't been in any of the previous trials by commercial makers. We want to open up to patients who have resolved CNS strokes and silent strokes, but who are not on chronic transfusions for stroke prevention. And we're going to add mortixafortide as a second mobilizer in cases where patients are resistant to mobilization. And we've now licensed successfully to Caring Cross. We hope to begin trials in Brazil, South Asia, and potentially Saudi Arabia in the future. And the key here is that we'll move to point of care manufacturing, which we hope will reduce costs significantly. And that's the vision of Boro and Caring Cross. And we've also been testing with reagents from XLthera, addition of UM71 in the manufacturing process in a hope to reduce genotoxic stress, which occurs at any time you take the bone marrow cells or the stem cells and put them in culture. So overall summary is we've introduced shmiR technology. It appears to be safe and effective. There's no genotoxicity. The cell manufacturing has been very consistent. Cell targets are efficiently attained in all the patients, most treated within months versus commercial products currently where there's waits. We have patients waiting very long time to get their product. The pivotal 2 multi-center trial has now reached its accrual target. And there is a need, I think, for additional study of genomics in this group of patients, and that's going on through several grants and several institutions from the NHLBI. There's multiple process improvements that we may introduce further to increase efficacy. These may be applicable to other ex vivo approaches, and I've mentioned already that we plan to expand access. We're very excited in our discussions with the FDA that they're likely to be amendable to those IND amendments, so we haven't submitted them yet. So let me acknowledge, first of all, the many patients. We treated now 36 patients altogether with this vector, and someone had to go first, so the patients community has embraced this. There's been a huge number of people involved at Children's Hospital. I'll just point out Erica Esrick, who was the PI, site PI for both the phase 2 trial and the phase 1 trial. The trans lab now called the TIL lab, led by Miriam Armand. We've had great collaboration with Bluebird. Now it has transformed to Genetics Biotherapeutics, a new name. John Manis, who's an absolute wizard at therapeutic apheresis. And we've had collaboration scientifically at the MGH and at the Institut Mondeur de Research Medica in Paris. And the funding has been through NIH for all of this, and CIRM I mentioned already. And our gene therapy program here is quite advanced. You can see a large number of people involved. And this was started about 15 years ago. We've done probably now over 30 human trials in gene therapy. And as you know, several of these are now approved by the FDA and are commercially available.
Thank you very much, David. That very impressive clinical data, and we're certainly looking forward to the phase 2 reveal of the data when it matures. So before we get to the questions, just a little bit for the audience about the partnership between Caring Cross and Boston Children's and Dana-Farber. Caring Cross has licensed this technology that you've seen from Boston Children's and Dana-Farber to enable its broad access to underserved populations, including those in the US and in low-middle income countries. And we'll do this by reducing the cost, which will be done by 2 major ways. First, we'll produce the vector at very high titer using Vector Biomed's proprietary lentiviral vector manufacturing technology here in Gaithersburg. And secondly, we'll collaborate with our global nonprofit partners like those in Brazil and India, who will use their local skilled technical labor to manufacture the final stem cell product locally at a much lower cost than here in the US. It's very well known that labor is a big chunk of the cost of producing these therapies. And Caring Cross will provide, licenses to its growing list of partners. And certainly if there's anybody here on the call that wants to learn more, that you can contact us through our Caring Cross website. So now we'll go on to our questions. What is the minimum effective dose?
Yeah, that's a great question. So in our phase 1 trial with 10 patients, we had 1 patient who we considered by protocol to be a failure. That patient, for reasons that we didn't understand, had a vector copy number in vivo of 0.2 and had suboptimal. She had induction of hemoglobin F, but less than what we would like. She's better overall, but she continued to have VOEs. So we assumed from that 1 patient that we needed in vivo VCNs above 0.2. And in the phase 2 trial, I think everybody had an in vivo VCN above 0.4, 0.5. So I think it is true, we've done a lot of ex vivo gene therapy trials. It is true every once in a while you run into a patient who, for reasons that we don't understand, doesn't engraft as effectively with the transduced cells, and that patient was one. But it was very helpful to us to improve our manufacturing so that we had a higher VCN in the drug product, which occurred in the phase 2.
What do you know about the stability of the shmiR within the cell? Do you have to fine-tune its expression level? Could you discuss the value of using lentiviral delivery of shmiR versus gene editing knockdown of BCL11A?
Yeah, it's all good questions. The shmiR is completely stable, so we've now followed patients out, as I said, over 6 years with the VCN, both in stem cells in the bone marrow and in the peripheral blood, completely stable. So I think that was a big question, of course, for us as we went into the phase 1 trial, since it had never been used in humans. So we're now satisfied that the vector, once inserted, is both stable in the genome and doesn't get transcriptionally silenced. The only way, you asked a good question about fine-tuning the expression. The way the expression is fine-tuned is by VCN. And so the knockdown efficiency and the induction of fetal hemoglobin is correlated with the VCN. And that's handy to have as a way of dialing up and dialing down the VCN. And then the last question, I think both of these approaches work. I think for us and for Karen Cross, the advantage of the shmiR technology is we don't need as many cells to be collected because the transduction with the lentivirus is not toxic to the cells. We have very good manufacturing efficiency, if you want to call it that. And in fact, in the phase 1 study, out of 10 patients, we collected enough cells for manufacturing with one mobilization cycle, 9 out of 10 patients. And as opposed to many cycles that are required for the gene editing often, not with everybody, but because the electroporation that you use for gene editing is toxic to the cells, of course. And then I think if you look at the phase 2 data I showed on hemoglobin F levels and fetal hemoglobin containing cells, that's at the level that gene editing is reporting. So I think both work, both have advantages and disadvantages. And I think for us, the manufacturing is and the ability to collect enough cells relatively quickly is an important aspect, because each time you do a mobilization cycle, you require 4 weeks in between for kind of recovery. You have to have a central line. Some places that requires an admission, so there's a lot involved if you have to do multiple collections.
Thank you for the great talk. Based on the current clinical trial results, do you think that this treatment could work for all patients of different ages?
Yes, I think that's the truth, that's the hypothesis. For the expanded access, we will reduce the age from age above 12 down. The thing about the age is that as you get lower than age 12, you may know this, the outcomes for allotransplantation improve the younger you are. And so I think there'd be a real decision people will want to make between an allotransplant versus gene therapy. But theoretically, it should work at any age, basically.
As you plan to change vector production, will new regulatory approval be needed, such a new IND? And as you plan to enter other countries, do you anticipate issues with regulatory approvals?
It's a good question. The vector production itself was done in a commercially viable process, but basically Boro and Karen's Cross job in part is to transfer the vector into their GMP production facilities. That will require, of course, FDA approval subsequently. With respect to the other countries, because the phase 2 trial was so robustly. I would say the oversight was audited in so many ways and robust. I think it'll stand scrutiny for regulatory review in other countries. I mean, that's yet to be tested, but that would be our hypothesis.
Yeah, I'll just add in terms of vector production, we'll have to look at the critical quality attributes between the existing vector and the production, and maybe do some, bridging studies or comparability studies. But I believe that the vector quality will be very, very similar. As you move the approach to ex-US, are there any concerns about different beta haplotypes of African and those in the diaspora, African Americans versus those on African continent, which is already known to have different phenotypes, hemoglobin F expression, and other genetic factors?
Yeah, it's a great question. I think from our limited data so far, we do see a difference in hemoglobin rescue in patients that have co-inherited alpha thalassemia trait and sickle cell. I think this is something also Bluebird has reported. So that's one consideration. But unlike potentially gene editing, this technology should be relatively indifferent to other haplotypes because the induction is using cell cycle or cell machinery as the switch, and the switch from fetal to adult hemoglobin is universal, no matter where you're at in the world. So we think this will be applicable regardless of the genetic background, with that caveat, which is I think that the phenotype in alpha thalassemia trait may be that you don't get rescue all the way up above 10 grams of hemoglobin, which is what we see in other patients, but you still will potentially get rescue of the vaso-occlusive crisis. So that's the one caveat, and that's based on very limited number of patients, obviously. So we'll need to do more exploration as we go forward.
If the transcription is successful with cells from one cycle of mobilization, what is the role of UAM171 in your collaboration?
Yeah, that's a good question. First of all, it's not included at this point. We've done and published on this in a preclinical setting. What we've noticed in the actual ex vivo manipulation of cells is that there's evidence of some DNA damage, and the use of UAM71 reduces that measurably. Now, that DNA damage has not been a problem with respect to clinical outcomes, but on the other hand, we think if we have a simple way to reduce that even further, that would be a good thing to do in the future. I think Cellular is being approved for ex vivo expansion of cord blood cells, and so it may already have FDA approval as an additive ex vivo, and therefore would be relatively easy to include in the future. But our plans discussed with Boro previously is that we're not going to change anything as we institute this into other countries in the beginning, because we want the regulatory agencies to feel confident that we're using the same methodology as we have data on for the 36 patients here in the United States.
Great, thank you. And Sarah Tariq asked a question: will the lentiviral vector work on everyone? Is there any restriction on using the lentiviral vector in any way?
Yeah, I think I previously touched on that. We don't see that at this point. We don't see any restrictions at this point. Yeah,
great. And I have one question just to finish up. Targeting the BCL11 locus with the shmiR and comparing that to overexpression of the beta globin. You touched on that in your seminar, but maybe you could expand on that, on the advantages and disadvantages of these approaches.
Yeah, so that's the Bluebird approach of expressing a modified beta globin gene. That works. That's got FDA approval for sure. I think that the theoretical difference is that in that approach, you're adding more globin genes, so you're expressing more beta globin, but you're not necessarily reducing fetal hemoglobin. You probably are doing it, but not by a binary switch. And that overexpression of the beta globin locus, if you think about it, without concomitant change in the alpha globin expression, could lead to an imbalance between beta globin and alpha globin, which is a definition of a thalassemia phenotype. Now, I don't think that's been a problem in their studies at all, so I'm talking more theoretical than not. I think another advantage is that our maximum induction of fetal hemoglobin occurs with an in vivo VCN of 1. So 1 copy of the vector in a cell, as opposed to multiple copies. And again, theoretically only risk of more copies in a cell of the genome is insertional mutagenesis. And I'm saying again, they haven't had that issue. I'm not saying that they did. I'm saying this is a theoretical advantage where we get maximal induction with basically a VCN of 1 in cells.
Given the favorable safety profile, how do you see implementation in patient populations with significant in-organ damage from long-term sickling events? Are there any practical or logistic hurdles?
I think the practical and logistic hurdle is that older patients, because of marrow damage, tend not to mobilize as well as younger patients. We will go up higher on our expanded access of the FDA. That's just a higher age, but the practical outcome is those patients will have a harder time mobilizing. And I think there is, again, more theoretically that the older you get with sickle cell disease, the more you may have evidence of clonal hematopoiesis. And so we will likely include in our expanded access for older individuals looking for clonal hematopoiesis as part of the entry into the trial. We're still in discussions about that, but that's the practical, I think. And as you're implying, but just to state it, there's no reversion of permanent organ damage. It's just stopping progression is what we're looking for.
Different countries have different comfort levels with conditioning. Are you thinking busulfan will be used everywhere? Is there an opportunity to change this?
Yeah, I think we will start out with Karen Cross picking sites that are used to doing allotransplantation and therefore used to doing ablative chemotherapy, and busulfan is the chemotherapy we'll use in the beginning. Of course, the field is all looking for the ability to do conditioning without chemotherapy. And if that happens and evolves, then I'm sure we'll be a fast adapter of that. We are working with other companies ourselves to try to develop that technology.
Great. And so thank you, David. Thank you for that presentation and the outstanding clinical data. We're very excited to be able to collaborate with you in order to improve access. And so I think we'll wrap this up. And I wanted to thank everybody also for participating on this virtual seminar. And I wish you all a great evening or morning or wherever you are. So thank you, everybody.